Good morning everyone. I hope you all are fine and doing well. Now in the last class we had discussed types of pollination and the agents involved in pollination, in cross pollination. So today I will be dealing with the agents and the types of pollination. So before I go into the detail of each kind of pollination, let us quickly go through the agents and the type of pollination. I had told you the agents can be abiotic, non-living, like wind and water. The type of pollination that occurs through wind is called anemophily, and by water is hydrophily. The other group of agents can be biotic or living. If insects are helping in pollination, we call it entomophily. If birds are helping in pollination, we call it ornithophily. If bats are helping in pollination, we call it chiropterophily. If snails and slugs are the agents of pollination, it is called malacophily. And if man is bringing about pollination, that is called artificial pollination. So we will now go into the details of each of these kinds of pollination and the characteristics which the flowers possess for that kind of pollination, for that kind of agent which is suitable for this. So we begin with the first, that is anemophily. Anemophily, I am going to erase this. Anemophily means pollination by wind, as it is indicated here. Now, anemophily is pollination by wind. This occurs in plants like coconut palm, in date palm, in maize, in cannabis. In Hindi, cannabis is called bhang. So in each of these plants, pollination is brought about by wind. Now, what are the characteristics of such flowers? First and foremost, the flowers have to be small. They are generally inconspicuous, means they are not very showy. They are small. They are sometimes green or of very dull colors. Now, the anthers of such flowers are long and they protrude out of the flower. That is, they hang out of the flower, so that when they are mature and are going to shed pollen grains, the pollen grains can be easily carried away by the wind. The pollen grains are very light. Pollen grains are light, they are dry, they are smooth, so that they can be easily carried away by wind. Now, the stigmas of such flowers are also large, long, they are feathery, and they also hang out of the flowers so that they can easily receive the pollen that is carried by wind. Now, when these plants are about to pollinate or are pollinating, the air is full with pollen, and pollen can be carried to thousands of kilometers away from where they are being produced. So these are some of the very important characteristics in flowers that are pollinated by wind. Now I will take one example and explain it to you, like maize. Now in maize, what happens? The male flowers are found at the terminal end, and and they are in clusters. The entire male inflorescence is at the tip of the plant. Suppose this is the maize plant. And in the lateral we see the female flowers. The feathery, silky, thread-like structures that we see are actually the stigmas. And the male flowers are produced in clusters at the terminal end. As I have told you, the anthers are long and they protrude out. So, the stigmas are large and they are feathery. So, pollen that are produced from the anther are carried by wind, will reach plants far from where they are being produced. Cross-pollination is being brought about by the agency of wind. These flowers are called anemophilous flowers and the pollination is called anemophily. Examples I have told you in coconut palm, in date palm, in maize, and in most of the grasses we see this kind of pollination. So this was one by abiotic agents, non-living agents. Now we come to the next kind of pollination, the second kind of pollination, what we call hydrophily. As the term itself tells you, this is done by water. Now we will discuss the characteristics that we see in hydrophilous flowers that bring about pollination by water. Examples of such plants are Vallisneria, Zostera, Hydrilla, and Ceratophyllum. These are some of the names of flowers in which pollination takes place by water. So these plants are found submerged in water. They are aquatic plants, they are found submerged in water. Now hydrophily has been divided into two kinds. First kind is called hypohydrophily. Hypohydrophily means the flowers are completely underwater. They are submerged underwater and the pollination is going to take place underwater. Inside water, the flower male part, the pollen grains, will reach the female part of the flower. This is called hypohydrophily. This is seen in Zostera and Ceratophyllum. The pollen grains, the characteristic is that these flowers are found underwater and the pollen grains are long and needle-like. And when they are produced from the anthers, they, with the help of water, reach the female flowers and they coil around the stigma. Coil around the stigma of female flowers and thereby they bring pollination, which will lead to germination followed by fertilization. So this is one example. Second kind of hydrophily is called epihydrophily. This is commonly seen in Vallisneria. In Vallisneria, the male and the female plants are separate. They are separate. Suppose this is a male plant. This is also Vallisneria, is also called ribbon weed. The leaves are like ribbon. Suppose this is the male plant. The male plant, the flowers are at the base. These are the male flowers. This is the surface of water. This is the female plant. The female flowers have a long stalk and the flowers are raised to the surface of water. This is a female flower. The stalk is very long. This is a female plant. These are the male flowers. Now when the male flowers are mature and the anther and the flowers are mature, the flowers break and they float on the surface of water. Suppose these are the male flowers. On maturity, they break from here and they float on the surface of water. And with water, they reach the female flower. And there they give out their pollen from the anther. Pollination takes place. And after pollination has taken place, this long stalk of the flower, it coils like this. And the female flower is submerged underwater. This is the mechanism of hydrophily, epihydrophily seen in Vallisneria. The fruit then develops. This is the fruit which later develops after pollination has taken place. The stalk coils and the flower is submerged underwater, which will develop into the fruit. So this is epihydrophily, the second kind of pollination brought about by non-living agents or abiotic agents. Now we come to the third kind of pollination, that is entomophy. Entomophy is brought about by insects. Now most of the flowers are pollinated by insects. There are many flowers that are pollinated by insects. Now insects basically come to flowers for nectar, for pollen. To take the nectar and to take the pollen, the insects visit flowers. Now insects like bees, butterflies, moths, let us say wasps, flies, and many more kinds of insects visit flowers for pollen and nectar. And when they visit the flower, accidentally they take the pollen with them, and when they visit the next flower, they drop the pollen there. But for an insect to visit a flower, the flower should have the following characteristics. The flowers should be large, they should be showy, they should be of bright colors so that the insects are attracted towards them. They should have copious quantities of pollen, should have nectar, should have fragrance, sweet smell, because of which the insects are attracted towards them. Now, the position of the anther and the stigma is such that when the position is such that when the insect visits a flower to take its nectar, the pollen are shed on the body of the insect, and the pollen grains are spiny. The pollen grains are spiny so that they can attach to the body of the insect. They are sticky so that they can attach to the body of the insect. So when an insect visits a flower, the pollen will fall, will stick to the body of the insect, and when it goes to another flower to suck nectar, to take the pollen, those pollen that were already present on the body of the insect will be shed at the stigma, shed on the stigma of that flower, and that's how pollination is brought about. Now, in some cases, it has been seen that there is a very close relationship between the insect and the plant. Like the tallest flower, Amorphalus, this flower is about 6 feet tall. The insects lay eggs in this flower so that their eggs can be protected, and thereby they bring about pollination. Then in another flower, Yucca, which is pollinated by moth. The moth also lays eggs in the ovary of this flower. The moth is the pollinator, it lays eggs in the ovary of the flower. And when it visits the flower to lay the eggs, it carries the pollen with it. When it goes into it, fertilization occurs, and as the larvae from these eggs are formed, the seeds that are produced also germinate, and that's how dispersal takes place. So this is very close relationship between the insect. Then another feature that has been seen, that in flowers that are produced at night, the flowers that are produced at night are pollinated by moths, because moths are nocturnal. They are like butterflies, but they are seen at night. So these flowers that are produced at night are generally white in color, and they have a very sweet smell to attract the insects, like queen of the night, रात की रानी, उसकी खुशबू बहुत ही अच्छी होती है. So because of that sweet fragrance, the insects, the moths are attracted to it, and in return they bring about pollination. So this was pollination by insects called entomophagy. Then we come to the fourth kind of pollination, that is pollination carried by birds, which is called ornithophagy. Now small birds like the sunbird, like the hummingbird, then honey thrushes. These are names of birds which are very small in size. They get attracted towards big flowers because they feed on the nectar of these flowers. And when they visit a flower, go inside the flower, they carry the pollen with it. The pollen sticks to its legs, to its feathers, and when it moves to another flower to suck nectar, they deposit the pollen on the stigma there. That is how pollination is brought about. So this is done by very small birds. Then this was the fourth kind. Now we come to the fifth kind of pollination, which is chiropterophily, brought about by bats. Now bats are also nocturnal. They visit flowers at night, either to eat the petals or to eat moths that are there on the flowers at night. Some bats feed on the flower parts, floral parts, like the petals, and they also feed on moths. So when they visit flowers at night in search of food, accidentally they bring about pollination. So, and as I told you, man is an important pollinator, and when human being bring about pollination, there is a specific purpose for it. They are trying to bring the desired qualities of two different types of plants into one. Like I was talking on the previous turn, that disease resistant varieties and high yielding varieties. The two characters brought together by humans by the process of artificial pollination. We also call it hybridization. So this is how the different agents bring about pollination, cross-pollination. Now after pollination, we will discuss the pollen-pistil interaction. Now when pollination takes place, the pollen reach the stigma of the carpel. All those events that take place after the pollen grain have landed on the stigma till the pollen tube that is produced on germination by the pollen grain enters the ovule are together collectively termed as pollen-pistil interaction. Now we all know, I'm just showing you a structure of the carpel. We all know a carpel has a basal swollen ovary, a slender style, swollen ovary, and the flat expanded portion, which is the stigma. Now on pollination, whichever way pollination occurs, the pollen reach the stigma. These are the pollen grains that have reached the stigma. So basically pollination is only transfer of pollen grains from the anther to the stigma. Now whether they are compatible, that is they are of the right kind, belong to the same species, or they are incompatible, of the wrong type, do not belong to the same species, that the stigma will be able to understand, because there is no guarantee that the right pollen have reached the stigma. So stigma is capable of recognizing the right kind or the compatible kind of pollen grain, and only if the pollen grain is compatible it will germinate. Now coming back to the structure of pollen grain, we had studied that pollen grain has two layers. The outer rough is the XII and the inner smooth is the intine, and the break between the XII forms germ pores. So these are the germ pores, and the pollen tube emerges from, it is an extension of the intine which emerges out through the germ pore, and it is a pollen grain has got two nuclei, the generative nucleus and the tube nucleus. Two nuclei are there. One is the generative nucleus, the other is the tube nucleus. Tube nucleus helps the pollen tube to move further. So, and the generative nucleus then divides to form two male cells or two male gametes. So now the pollen tube, after the right pollen has reached this, several pollen grains will germinate, but only one of them will be able to move down. The pollen tube will extend to reach the ovule. Now coming back to the ovary, the ovule is present inside the ovary. It contains the embryo sac surrounded by the nucellus. Two integuments cover the embryo sac, the outer integument, the inner integument, and by means of funicle it is attached to the wall of the ovary. So this is the egg cell. This opening of the ovule is the micropyle. So the three cells, embryo sac is made up of 8 cells. Embryo sac is the 8-cell type. So the 3 cells towards the micropyle, one, the central one is the female gamete or the egg cell. The other two are called synergids, and in the center there are 2 polar nuclei, and these polar nuclei then fuse to form the secondary nucleus. And this end of the ovule is called the clasa. And 3 cells here are the antipodal cells. I have explained this, which we have done earlier, to you once again, because now when we study how pollen tube germinates, and once the male gametes reach the embryo sac, how fertilization occurs. So pollen tube develops from the intine and it enters the ovule through the micropyle. There are 3 ways in which the pollen tube can enter. 3 different ways in which the pollen tube can enter into the ovule. In most of the cases we see that this pollen tube elongates and enters the embryo sac through the micropyle. Through the micropyle. The pore is the micropyle. This is called porogamy. Entry of the pollen tube through the micropyle into the embryo sac is called porogamy, which is the most common method of entry of pollen tube into the ovule. Another method is if it enters directly through the clasa. Through the clasa, if the pollen tube enters like this, through the clasa and then reaches the embryo sac from here, then that is called clasogamy. This is seen in Casuarina. And the third is when the pollen tube enters through the integuments like this, germinates from the stigma, moves through the tissues of the style and enters the ovule through the integuments is called mesogamy. This is seen in cucurbits. Now, how has pollen germinated? The stigma recognized the right pollen, the compatible pollen. Pollen absorbed water, sugar, lipids produced. This is called the stigmatic fluid, which is produced by the stigma. It absorbs that. Then it germinates. That is the pollen tube. The intine emerges as a cytoplasmic extension through the germ pore and carries the two male gametes. Tube nucleus is helping it to elongate, and the tip of the pollen tube carries these two male gametes, male cells or male gametes. There is an enzyme called cutinase present at the tip of the pollen tube, which is helping in dissolving the cells of the style to finally allow the pollen tube to reach the ovule. Three different ways in which it can enter the ovule. If it is through the micropyle, it is called porogamy. If it is through the clasa, it is called clasogamy. And if it is through the integuments, it is called mesogamy. So now, all these events that are taking place after pollination till the pollen tube has entered the ovule are grouped together as pollen pistil interaction. Now, once the pollen tube has reached the embryo sac, what is going to happen? That we will see now. That is termed as fertilization. And fertilization means fusion of dissimilar reproductive cells, male and female cells. They fuse, and that is called male gamete fuses with the female gamete, resulting in the formation of cells. Now, this was for the first time seen in angiospermic plants by Strasburger. Phenomena of fertilization was studied by the scientist Strasburger. Now, angiosperms show a special kind of fertilization which is called double fertilization, and this was reported by the scientist Nevaschil. Double means two male gametes are produced. Now, when the pollen tube enters through the micropyle, it enters the embryo sac through one of the synergids. It enters the embryo sac through one of the synergids, and in the cytoplasm of the synergid, it leaves the two male gametes. Now, these two male gametes, one male gamete will fuse with the egg cell. One male cell or male gamete will fuse with the egg cell. Egg cell is a haploid cell. This male gamete is a haploid cell. So, we get a diploid cell, and this diploid cell is the zygote. This diploid cell fusion, which has been formed by the fusion of the egg cell and the male gamete, is the zygote. This zygote will undergo series of divisions and will ultimately form the embryo. The second male gamete will come in contact with the two polar nuclei which have fused to form the secondary nucleus. Secondary nucleus is diploid. Why? Because it has been formed by the fusion of two polar nuclei. Now one more male gamete comes in contact with the secondary nucleus and forms a triploid structure. This is called triploid fusion or triple fusion. And this triploid cell is called the endosperm nucleus, which will result in the formation of the endosperm. What is the endosperm? Endosperm is the nutritive tissue which is going to provide nutrition to the developing embryo inside the seed. This whole ovule, after the process of fertilization, the ovule after the process of fertilization forms the seed, and the whole ovary after the process of fertilization results in the formation of fruit. All other parts, the style, stigma, calyx, stamens, petals, they degenerate. The ovule forms the seed, the ovary forms the fruit, and the fruit contains the seeds. And when we sow the seeds, the embryo forms the seedling. The seed germinates and the embryo results in the formation of a seedling. So this is what is double fertilization and what is triple fusion. Double means two male gametes, one fuses with the egg cell, other fuses with the secondary nucleus. How was secondary nucleus formed? By the fusion of two polar nuclei. Those polar nuclei is a diploid structure. After fusion forms the secondary nucleus, which is diploid, and one more male gamete fuses with that, which is called triple fusion, results in a triploid cell called the endosperm nucleus, and this endosperm nucleus will form the nutritive tissue endosperm. So this was pollen pistil interaction followed by fertilization. So I will end here today, and in the next turn we will move on to the next topic. Stay safe and stay healthy till then.